Hello everyone, and welcome to our online lecture series for Bio 2. Today, we hopefully visit a topic that you are at least moderately familiar with Punnett squares, and then spend the next couple lectures building on this understanding. So today is going to be a fair bit of vocabulary. It's going to be some drawing out of examples, some problem solving. I do expect that you are able to solve these problems alongside me as we are working on them, because come time the exam, you will be expected to solve these on your own. I do expect you to keep with the nomenclature that you're going to be taught in this class. I understand that you may have previous exposure to capitals meaning dominant and lowercase meaning recessive, but I want you to disabuse yourself of that understanding. We'll be using a different nomenclature that I'll explain, so please pretend like you're wiping the slate clean and make sure that you're using the system that we have in this course. All right, so Punnett squares are an expansion of our understanding of genetics. Genetics is the way in which we transmit hereditary information from parents to children. So that begins by defining the parental generation. We'll abbreviate the parental as P. The children of the parents are called the F1s, or the filial 1 generation. They're one generation removed from the parents. They are a result of the mixture of the gametes, or the sex cells of the parents, in order to make the appearance and the genetic makeup of that filial 1. The appearance of groups, or individuals, we call as the phenotype. How they look. Underlying the phenotype, we have a genotype. The underlying alleles that are responsible for determining the pheno, or the phenotype. So, in some way, your genotype can be used to predict your phenotype. This prediction can go beyond the F1, or filial 1, to the F2, which is our filial 2, or our grandchildren of the parents, or even the filial 3, the great-grandchildren, and so on and so forth. We can really build this back. Now, of course, we've talked about these terms before, but let's make sure that we have them solidified for our Punnett squared discussions. We know that when talking about a genotype or a particular underlying genetics, we are talking about the gene that underlies the trait. That gene has a locus or location on some chromosome. And what we care about in determining how genotype predicts phenotype are the different alleles that individuals carry. Now, we know that genes are found on certain locations on the chromosome. Let's go ahead and say, oops, I shouldn't use blue, pardon me. We're talking about a gene at the very end of the chromosome. It is found on each of these chromosomes, chromosome 1, in these 4 gametes, which are the product of recombination and segregation and independent assortment. So, reviewing those terms here, we know that recombination means that in some way we're able to mix and match the different allelic combinations. So this allows you to mix and match alleles that are found on the same chromosome. We also know that alleles on different chromosomes will be completely separate from one another, thanks to the law of independent assortment. Oops, do not get carried together. And then by the law of segregation, we know that mom and dad alleles separate. Oops. And so that's fundamentally what we learned in our meiosis discussions, in our reproductive discussions. We learned to look at these alleles as they're moving into their gametes. And we learned how to draw them, arriving into these different sex cells. Now we're going to get rid of this segregation into gametes and shortcut it by drawing them into Punnett squares. But I always like to start this section with a bit of understanding of how do genetics work? What's the basic idea of how alleles interact? Well, originally, in the before times, so before the scientific revolution and our understanding of genetics in the very, very distant past of, the 1900s and mid-1800s. So these ideas are relatively young in terms of human history. Oh, before times. Sorry. Folks certainly understood that there was something to being a parent in determining child phenotype. For example, if two parents mated and they both had black hair, it was highly likely that their children would have black hair. If one parent was tall, one parent was short, the child would be likely something in between. Therefore, our original Greek, Roman, and medieval understanding of genetics was that blending occurred in parentage. That each parent contributed equally, and the child would be some average of the two parents. Well, we know that that's definitely not the case. We know that some traits don't really have a medium, right? Like eye color. We know that some traits are inherited as like a flip of a coin, like gender. Male or female, biologically here. We know that we don't all tend to some average, because think about it. If genetics was equal blending, contributing equally, that would mean that after some number of generations, every single human being on planet Earth would look exactly the same. And that's definitely not the case. We all look slightly different, still all being Homo sapiens. And our diversity is fantastic. Genetic diversity is a good thing. So we know that over the long term, this fails. So we waited, as a species, quite a long time to get to a point where we were able to get a more detailed understanding of genetics. And the individual who led this revolution was a monk by the name Mendel. So Mendel was a monk born in 1822, died in 1884. So we can give him a time frame of the mid 1800s to give us a ballpark of when he was doing his thing. He was born in what is now the Czech Republic, though at the time it was a German-speaking province in the Austrian Empire. He did a lot of good stuff in his life. I actually encourage you to read about his life since he did so many interesting things. And of course, his major contribution was with working with pea plants. Latin name, Pisum sativum. Oops, sorry. Pisum sativa. Wow. The common pea. He worked with 7 different characteristics: plant height, pod shape, pod color, seed shape, seed color, flower position, and flower color. And with these 7 traits, he was able to show the basics of modern genetics. His idea was to get a plant that was true breeding for a trait. Like, my personal favorite here is seed color. So true breeding for seed color. In his population of plants, he noticed that there were peas that were yellow in color, and there were peas that were green in color. And in order to be a true breeding plant, what that meant was that a yellow pea-colored plant would cross with itself, and always the F1 generation would be 100% yellow. Same would apply for a true breeding green plant. It would cross with itself, and the F1 generation would be 100% green. So 2 questions from this. How do you self? Well, plants are, in many cases, both genders. A single individual will make both sperm and egg in a single perfect flower. This allows you to take pollen from that same individual and cross it to itself, put it on its own female eggs, and fertilize itself. That's what we call selfing. Second, what does this mean for the underlying genotype? Well, for a true breeding plant, that means that it has 2 alleles, and if it's a true breeding yellow, it's going to have 2 yellow alleles. So now I'm going to introduce you to the method of, let's go ahead and move this up so I have a little bit more space here. For a true breeding plant with 2 alleles, you need to be able to indicate these different alleles. For every exam question or notes or everything from here on out, I want you to follow this naming system to make it very clear to me what is your nomenclature naming system. So you are always going to give a gene letter. And in this case, if you want it to be seed color, you can go ahead and use the letter S equal to seed color. That is our gene. In order to indicate our alleles, you're going to write S with the subscript G. This will be our green color. And S with the subscript Y will be our yellow color. Notice, no capitals, no lowercase, no implicit understanding of dominant and recessive here. These are just different alleles. This will help long term. It seems a little bit of a pain in the butt, but it will help long term, I can promise you that. So if every single parent that's true breeding has two alleles, you know that a true breeding green plant will be SG, SG, because either of its allelic combinations have to match to give a fully green plant. We don't know whether green is dominant to yellow, or we don't know whether yellow is dominant to green, but we do know that an individual that's true breeding has to be homozygous. In this case, this individual is homozygous green. You can also be homozygous yellow. Both of these are true breeding because the egg will get to, let's make sure to colors here. The egg will get to pick one of these two alleles, either an SG or an SG. The sperm will be made from one of these two alleles, an SG or an SG, and therefore the only possible combination is an SG, SG. True breeding. If instead the individual was SG, SY, we don't know whether one is dominant or recessive yet, but they could either pass on the SG allele or the SY allele. And in this case, you could make progeny that were SY, SY from selfing this individual. You can make progeny that were SG, SG, or you can make progeny that are SG, SY. An individual that produces a plethora of different progeny has two unmatching alleles and is therefore heterozygous. So homozygous alleles match, heterozygous alleles do not match. So then how do we figure out dominance and recessive? Well, a gene that is dominant not gene. Only alleles are dominant or recessive. Because remember, the gene is just something like seed color, and your alleles are either green or yellow. So you need to figure out whether green is dominant or whether yellow is dominant. The dominant allele will mask the other in a heterozygote. And I will abbreviate heterozygote as het from here on out. Het and hom, or homozygote. So, the way to determine dominance is by looking at the phenotype in a het. And so now I'll tell you that in an SG, SY individual, the phenotype is green. Whoop, green's definitely got two E's. This means that the green allele masks the yellow allele, so green is dominant, yellow is recessive. Pretty straightforward, but keep in mind that in order to understand dominance and recessive, you have to be able to analyze the phenotype in a heterozygote. Once we have this understanding of dominance, recessive, heterozygous, homozygous, we can start building it out to more complex crosses. For example, a cross between two true breeding pea plants with different flower colors, violet and white. When you cross this one to itself, it's 100% violet. That means if we're going for flower color, the gene F, color of violet, color of violet. This is the genotype for this particular individual. In our white individual, flower color is white, flower color is white. Of course, on the exam, I want you to be able to, and in your notes, write out that the gene name is F for flower color, FV is equal to violet color, FW is equal to white color. And so let's take this parental generation and cross it. This, because you are crossing two true breeding plants, is known as a hybridization. The F1 hybrid has violet flowers. Now you know that the F1 received either the FV allele or the FV allele from one plant, or the FW allele or the FW allele from the other plant. That means you know that it has an FV and an FW. This means that FV is dominant to FW because the phenotype in the progeny is purple. This is how you can indicate dominance and recessivity. So let's go ahead and self this generation. Well, now you know that there are two different possible alleles. In order to illustrate how this works, the best way to do so is with a Punnett square. In a Punnett square, one axis is the side for your maternal alleles. The other axis is the side for your paternal alleles. When you are dealing with one gene, you are going to get a box that is 2 by 2. Because mom can donate one of the flower alleles, flower color alleles, and dad can donate one of the flower color alleles. So that way the contents of every single box in this Punnett square is a diploid individual. Meaning that this is the possible egg, and this is the possible sperm that are made. We're not talking about total counts here, we're talking about the possibilities. We assume that there is a 50% chance of making this and a 50% chance of making that egg. We assume that there is a 50% chance of making this sperm and a 50% chance of making this sperm because of the law of segregation. Now looking at that hybrid, this individual here that we are selfing, we know that one of the alleles that the egg can receive is a violet, and we know that the other allele that the egg can receive is a white. For the the same thing goes. A violet and a white. Now, it does not matter whether you listed the white or the violet first. You can flip them around. Ultimately, the counts at the end will be the same. Now, we know that violet is dominant to white. So let's go ahead and fill in this box with the respective genotypes and then observe the phenotypes that we get. Well, here you're going to make an individual that's FV, 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 FW, because I am carrying one allele over. So you've got the V from here, the W from here. Same thing here, you're going to have the V. Oops. I'll write it out then, indicate it. FV, FW, does not matter the order that I write them in. FW, FW. So, our genotypes at the end of this cross are 1 F, V, F, V, 1 F, W, F, W. So there's our FV, FV, there's our FW, FW, and then 2 FV, FWs. This individual ends up looking violet, this individual ends up looking white, and these individuals end up looking violet. This means that you get a ratio of 3 violets to 1 white when you perform these crosses. And now, the benefit of doing this in plants rather than humans is that these crosses can be done hundreds of times. So in the F2 generation of close to 1,000 babies, you're seeing a ratio of 700 to 250, or 224, 705 to 224, violet to white. This has an approximate ratio of 3 to 1, based on your Punnett square. But of course you know that in true random sampling, you're not going to get every single one of these babies made every single time. You're instead going to get a random draw from this box. That means it's similar to flipping a coin. The ratios will not always be perfect, but they will approximate that value. All right, so that's one example. Let's walk it through again with yellow color and green color on peas. Note, however, that your book does not abide by my naming convention, which, sure, but we'll keep in mind that this trait, looking at a monohybrid cross, because you're looking at a completely yellow and a completely green pea color here, they should be labeled, for being consistent with my nomenclature, P equals pea color, and PY equals yellow, PG equals green. Now, your book illustrates it showing that in the parental generation for each of these true breeding plants, where one is true breeding for yellow, homozygous yellow, the other is true breeding for green, homozygous for green. The only gamete that they can make is either the PY or the PG. However, we know that when they mix, the offspring, the F1, will be heterozygous. It can produce 2 different types of gametes. When looking at either the female or eggs or the sperm, please note that now and forever, this symbol indicates female or egg. This symbol indicates male or sperm, shorthand for them. We know that the self-pollination produces F1 offspring. Self-pollination of the F1 offspring produces F2 in a ratio of 3:1, based on this Punnett square. And then we can see the ratios of the genotypes and the phenotypes at the end. So you will be performing these ratio comparisons throughout your different example problems. All right, so hopefully that clarifies how to do a cross of one gene with two alleles in the F1 and the F2. Note, in both of these, I was asking you to do a monohybrid cross. I could additionally ask you to cross, say, a yellow pea and a yellow pea that's heterozygous. Please take a moment to calculate the ratios there. All right, so let's do it. Well, doesn't matter which one's male, female, doesn't matter which one's egg, sperm. You know the ratios, or you know the alleles that are being given. This individual here would be PY, 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 PG, PY, PG. Oops. All of these are going to be yellow. But one half of them will be heterozygous, one half of them will be homozygous, yellow. And so in this way, you should be able to fairly quickly come up with these different ratios given different crosses. Now, looking at all 7 of Mendel's traits, since we'll be mixing and matching for our next little exercise, we have flower color, violet versus white, flower position, axial or on the side versus terminal or on the end point. We know that the F1 offspring are 100% violet and 100% axial in these 2 different traits. I won't read through all of the traits, but you basically get an idea that the F1 is where you're going to be determining dominant recessive. And then you see the reemergence of the recessive phenotype in our F2 offspring. Now, these are Mendel's exact numbers that he published in the mid-1800s that were rediscovered in the early 1900s to then give us a foundation for genetics as a field. And we can see, that these ratios are captured in the F2, showing the roughly 3:1 ratio in all of our different true breeding, parentals, hybrid F1s, and mixed F2s. So what do we do with this? What kind of problems can we solve? Well, one type of question that you might be asked or on the exam or in your own life, if you're going into a career as a genetic counselor, would be to look at either the product or the sum rule in terms of 2 different events and their relationship to one another. Because, for example, let's say that we were looking at the likelihood in an F1 offspring cross of these 100% violet flowers, where the parents were true breeding, what's the likelihood of getting a flower that's violet? And then from a second cross, getting a flower that is axial. Now these are 2 totally different gene pools. You don't know the flower color here and you don't know the flower position here. So you're just completely separate here, 2 independent trials. Let's go ahead and write out these ratios. Well, if I'm looking at, oops, sorry. I will do flower color as a C with a V or a W. I'm shorthanding it here, but please do not do so on your exams. And flower position as either axial or terminal. Now, flower color as violet, white. Oops. Flower color as violet or white. This individual would be violet. This individual would be violet. This individual would be violet. This individual would be white. So therefore, you get a ratio of 3/4 violet to 1/4 white. On the flower position, we know that they are axial and terminal. Axial and terminal. Keep in mind, this is the cross of the F1s, and this is also the cross of the F1s. So this is a very specific genetic problem that I'm giving you. In this case, axial, axial, axial terminal, axial terminal, and terminal terminal. Now, I don't remember which one of these was dominant or recessive. So in the cross of axial versus terminal, I see that axial comes up. That means I know that axial is dominant to terminal. So you're getting 3/4 axial, 1/4 terminal. So, what is the likelihood from these two different crosses if I told you to pick up if you randomly grabbed one, so you cross this F1 and pull one progeny, and you grab this F1 and you grab one progeny, what is the likelihood that the flower will be violet and the other flower will be axial? So you'll notice that I used the term "and" between these. That should hint you towards the product rule, as these are two independent pools. You've got one pool from your color, one pool from your position. And since the probability of pulling a violet flower is 3/4, and then the probability of pulling an axial flower is also 3/4, the probability of getting both of these occurring in a single event is going to be the multiplier of this. And 3/4 times 3/4 is 9/16. Or if you prefer using a calculator, 0.5625. So the likelihood of pulling a violet flower from this group and an axial flower from this group in two independent draws is 56.25%. We can use the product and sum rules in another, perhaps more familiar utilization. I am one of 3 boys in my family. So that means you have 3 children. We were 3 boys: Mark, Paul, and Chris. What was the likelihood for my parents to have had 3 boys? Well, you had a 1/2 chance of pulling a boy the first time, 1/2 chance of pulling a boy the second time, 1/2 chance of pulling a boy the third time. Since these are 3 independent draws, 1/2 times 1/2 times 1/2 is 1/8. Now, you could look at a slightly different question here. Instead of asking what's the likelihood of having 3 boys, you could ask the question of what's the likelihood of having 3 children of the same gender. Well, you could get 3 boys, or you could get 3 girls. Notice the use of that or there. What is the likelihood of having 3 boys? Well, it's 1/8. What's the likelihood of having 3 girls? Well, it's 1/8. So what's the likelihood of having 3 boys or 3 girls, meaning 3 children of the same gender? That's where we have to use the sum rule. 1/8 plus 1/8 gives you the likelihood of 2/8 or 1/4, or 25% chance of having 3 children of the same gender if you only have 3 kids. So hopefully this explains to you the places where we're using the product rule, independent events where you're looking at both of them occurring, or the sum rule, mutually exclusive. You can't have 3 boys and 3 girls if you only have 3 children. You have to look at them separately. So both will be fair game for our different methods of evaluation. Moving on from there, we will tackle the dihybrid cross. So first I'm going to show you how this works in a model from the book, and then afterwards I'll draw an example of this out on our own. So in a dihybrid cross, you again have a parental generation based on seed color of either CY or CG, and seed shape based on shape round or shape wrinkled. And again, all that they're doing here is selecting different traits from Mendel's assortment of dominant and recessive traits here. Today we're doing all simple genetics, mostly focusing on the math. In looking at this dihybrid cross, you have a parent that's true breeding for yellow, color yellow, shape round, and color green, shape wrinkled. They're using lowercase and uppercase here, but again, we can follow the nomenclature. The F1 is a hybrid, but it looks round and yellow. In order to do the Punnett square for 2 traits, because we are now looking at 2 genes, the gene for color and the gene for shape, our square will no longer be a 2x2. Instead, it will be a 4x4 if you're looking at all of these at the same time. Now, that means that you are going to be able to mix and match the genes and the alleles. So in the situation where you have color yellow, color green, shape round, shape wrinkled, you can use the FOIL rule to generate all of your different gametes. The FOIL rule states that you want to pair across your different genes, so you're never pairing a CY with a CG. That does not occur. You're instead pairing your CY with the first of the shapes. So the first possible gamete, CY, SR. Then you're going to pair the outers, or O for outers, CY and SW. Then you're going to pair your inners, or your CG and SR. Color green, shape round. And then you're going to pair your lasts. Your color green, shape wrinkled. And in that way, you have the 4 gametes generated by either the mom, either the dad or the mom. Now, in interest of keeping this a little bit quicker, I'm going to copy these. Keep handwriting consistent here. Make my own square. The square will be in black. 4 rows, 4 columns. Then, just like before, when you had a single trait, you need to carry each of the different genes into each of the different squares. So, looking at our box here, this individual here would be CY, CY, diploid for color, and diploid for shape. This individual ends up being yellow and round. Move to your next square, CY, CY, SR, SW. This individual is still yellow and round. Actually, let's I don't want to do this. Now let's just keep this nomenclature. So the 2 dominants there. You can do this for any one of your squares, filling in the remainder of this. So I will kind of quietly do so here. CY, CG. Please do the same at home. Again, I understand that this can be a little bit tedious, but you will be expected to be able to replicate this, and we will get more complicated the next class around. So it behooves you to train those arm muscles and do some copying. Going through the motions builds attention to detail. It builds the ability to keep track of a lot of moving parts. And it forces you to do this filling in of the square with diploid alleles many times over. So that way you don't do some of the more common mistakes on the exam, like filling in squares or filling in gametes that have 2 alleles from the same gene. I've watched that happen one too many times. It's not great. Whew, almost there. Those barks are going to stay in the lecture. I agree, Odie. Thank you. Good boy. He woke up from his nap to sound off there. Now he's looking at me like I'm talking to him, like I usually am with all the lectures. A very patient pup who's listened to all of them. And the very last square, CG, CG, SW, SW. So now, for our colors, let's do something similar to what the square above did, where the book and a graphic artist color everything neatly based on fill in shapes as well. I'll go ahead and color or highlight all of the squares that are yellow now. If it has a yellow, yellow is dominant, so you can ignore the fact that the overall genotype might be heterozygous. Now, of course, the other way to notice this is that this is a yellow and this is a yellow, so the entire rows will be yellow. And this is a yellow, or this is a yellow, and this is a yellow, so therefore these entire columns are yellow as well. Your greens will occur where you have 2 greens. So you'll notice that this is just like our smaller Punnett square. We have 3/4, or 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 possible yellows and 4 possible greens. This has that same ratio of 3:1, or 1/4 green, 3/4 yellow. Just over a bigger Punnett square because of the fact that we're working with the dihybrid. Now for our round and wrinkled, I'll draw our shapes. Again, we know that rounds and rounds are dominant, so we got our round, 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 round. Here's our round here, here's our round here, so let's go ahead and fill in each of these rows and each of these columns. Again, 12 round, wrinkle, wrinkled, wrinkled, wrinkled. 12 that are round, and 12 that are wrinkled. 12 that are round and 4 that are wrinkled. That's it. Gotta be paying attention to detail, making sure that your ratios are not off. But what we really care about is looking at how often these things occur. So you could go back to your Punnett square and be like, all right, 1, 2, 3, 4, 5. Whoop, nope, sorry. I'm counting. Let's start over here. I'm counting all of those that are yellow and round, and then crossing them out. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 yellow rounds. 2, how many yellowed wrinkles? 1, 2, 3. 3 yellow wrinkled. How many green round? Well, green round, green round, green round. 3 green rounds. And 1 green wrinkled. Whew! So you've drawn out the entire Punnett square here. Those of you who wish, and I recommend that you continue to do this so that way you have practice doing the dihybrid cross, but you'll note that you could also go about this using our product rules. If I do 2 separate Punnett squares, a CY, CG, cross a CY, CG, oops, you know that 3/4 will be yellow and 1/4 will be green. Same goes if I was to do the Punnett square for round and wrinkled. 3/4 would be round, 1/4 would be wrinkled. So, if you're trying to get the likelihood or the ratio that you'd expect for one of these traits, not all of them, you could use the product rule. What is the likelihood that a single P selected from a dihybrid cross would be yellow and wrinkled? Well, the odds of that are 3/4 yellow times 1/4 wrinkled, meaning that you're looking at a likelihood of 3/16. Same as you had up there. Another question. What's the likelihood of being yellow and round? 3/4 times 3/4 gives you 9/16, present there. So in this way, dihybrids are actually the product rule for two separate monohybrids. This still relies on the fact that you need alleles to segregate, meaning that you get yellows and greens going to separate gametes, but you also need alleles to sort independently. Yellows are not more likely to be inherited with rounds. You have a 50-50 chance of being yellow and round and yellow and wrinkled. Note that this goes all the way back to our independent assortment. Here are two chromosomes. This chromosome here is the color chromosome, has the color gene, and this chromosome here is the shape chromosome. Let's say that our dad allele is our wrinkled on shape, and our mom allele is our round on shape. And let's say that our mom allele on color is yellow, and our dad allele on color is green. Going back to meiosis, you see that there is a 50% chance that you are yellow and round versus an equal likelihood for these chromosomes to have lined up on the opposite sides of the metaphase plate for a 50% chance that you are yellow and wrinkled. So those two correspond to these two boxes. And so on for the rest of this. So hopefully you feel comfortable either drawing out the 4x4. I may ask you or obligate you to draw the 4x4. I may ask you to draw two separate 2x2s and give me individual calculations. Now, the 4x4 is easier if I'm asking you for everything. The two separate 2x2s are easier if I'm asking you for one particular phenotype. Now, I won't always be giving you dihybrids. You may get some loci that are homozygous, some loci that are heterozygous. Some genes that are homozygous versus some genes that are heterozygous. So be ready for any of the mix and match. We'll leave this lecture having completed our dihybrid crosses, but I want to make sure that before we move on that you understand these things incredibly thoroughly. Because next up, we change the inheritance patterns. We change whether they're heterozygotes or homozygous. And ultimately, we even change the gene interactions between our two different loci, meaning that one is not independent of the other. See you next time, but please make sure that you understand this system of inheritance through Punnett squares very confidently.